Welcome to Lecture 8 on soil and plant secondary macronutrients, calcium, magnesium and sulfur. This lecture is part of the Soil Science and Plant Nutrition subject ALM114, which is offered in the agricultural degree at North Melbourne Institute of TAFE. Please visit our website for further details about our courses at www.nmit.edu.au. This lecture was put together by myself, Dr. Nikki Cooley, and Dr. Doug Rao. Calcium, magnesium and sulfur are all secondary macronutrients. They are considered secondary macronutrients because they are less commonly yield limiting than the primary macronutrients, nitrogen, phosphates and potassium, which we covered in lectures 6 and 7. Yet they are required by crops in relatively large amounts. Although many soils in Australia and around the world have adequate secondary nutrients for crop production, sulfur deficiencies are unfortunately on the rise. This has been cited in regions of the US and throughout the world by Rasmussen and Krishij in 1986. Three global trends are responsible for increasing sulfur deficiencies: the shift in modern fertilizers to more concentrated, higher analysis forms containing little to no sulfur, a historical byproduct of the manufacturing process; the reduction of sulfur dioxide emissions from burning coal and oil when decreased atmospheric sulfur additions; and finally the steady increase in crop sulfur uptake and removal due to high-yielding varieties and more productive management. Effective management of secondary nutrients requires an understanding of the process that determined their availability to the crops and the methods to manage soils when you have inadequate secondary nutrient levels. Also, large amounts of calcium and magnesium are added when lime is added to acidic soils. This occurs frequently in Australia. Sulfur is usually found in sufficient amounts from the slow decomposition of soil organic matter, an important reason for not throwing out grass clippings and leaving leaves on the ground. In this lecture today, we are going to look at some detail of these macronutrients. We're going to examine their function in plants and the role they play. Why are they important? We'll have a look at some of the deficiency symptoms that they exhibit. We will briefly discuss their cycles, their poles, processes and fluxes. And finally we will look at some examples of some commercial fertilizers containing these macronutrients. Let us start by learning about the macronutrient calcium. Calcium is an essential plant nutrient. It has many roles and functions. It participates in metabolic processes of other nutrients uptake, for example nitrate uptake and metabolism. It promotes proper plant cell elongation and has an important structural role. It strengthens cell wall structure. Calcium is an essential part of the cell wall. It forms calcium pecitate compounds, which give stability to cell walls and bind cells together. It is the ability for calcium to produce strong cell walls that aids in disease. It can help resist fungi and bacteria that secrete enzymes, whose aim is to impair into the plant's cell wall. Calcium can impact on fruit quality. It has an important role in the regulation of stomata. Calcium also is involved in starch metabolism. Calcium is transported in the xylem via an ion exchange mechanism. It attaches to ligand molecules and exchange must occur with calcium or another similar cation, for example magnesium, sodium or potassium. Calcium is not very mobile in the soil or in plant tissue, therefore a continuous supply is essential. The relationship of calcium uptake and xylem links calcium uptake to transpiration. Calcium mobility and uptake in plants. Calcium uptake by the plant is passive and does not require energy input. Calcium mobility, as stated in the previous slide, takes place mainly in the xylem together with water. Therefore, calcium uptake is directly related to the plant transpiration rate. High transpiration rates mean high calcium uptake. Therefore, conditions of high humidity, cold, which produce low transpiration rates over a long period may result in calcium deficiency. Salinity buildup might also cause calcium deficiency because it decreases the water uptake by the plant. Since calcium mobility in plants is limited, calcium deficiency will appear in younger leaves and in fruits because they have very low transpiration rates. Therefore, it is necessary to have a constant supply of calcium for continued and optimal growth. Now let us talk a little about soil calcium content. The preferred level of calcium in agricultural soils is greater than 5 megaequivalent per 100 grams, or 1,000 mg per kilogram. There are certain soil characteristics that impact on calcium plant availability. Calcium forms insoluble compounds with other elements in the soil, such as phosphorus. Calcium that is in the form of an insoluble compound is not available to plants. Since calcium is positively charged ion, it is absorbed in the soil to the surface of the clay and organic particles which are negatively charged. Positively charged ions absorbed to soil particles are termed or called exchangeable ions because they can be exchanged by other ions present in the soil solution. Soil analysis determines the level of exchangeable calcium ions and not the total calcium in the soil, because the exchangeable calcium is the form which is available to the plant. Several factors or characteristics in the soil can assist in plant availability, and these include soil pH. Usually soils with a higher pH level contain more available calcium. This can be an issue in Australia as our soils tend to be more of an acidic nature. CEC is a soil characteristic that describes the total amount of positively charged exchangeable ions that the soil can hold. A higher CEC indicates a higher capacity of the soil to absorb and hold calcium, therefore higher calcium availability. And lastly, the presence of competing ions. Calcium competes with positively charged ions such as sodium, potassium and magnesium. Applying too much of these positively charged ions might decrease calcium uptake by plants. Sodium ions can replace the absorbed calcium, damage soil structure and decreases calcium availability. While calcium is an essential element for all plants, the following crops have been found to have especially sensitive responses to calcium. These include apples, broccoli, Brussels sprouts, cabbage, carrots, cauliflower, celery, cherries, citrus, conifers, cotton, the family of plants the cucurbits, melons, grapes, legumes, lettuce, peaches, peanuts, pears, peppers, potatoes, tobacco and tomatoes. If you suspect a calcium deficiency and you are growing one of these plants, you should take extra care to ensure proper identification. Calcium deficiency symptoms can be rather vague and therefore difficult to identify, since the situation is often accompanied by low soil pH. Visible deficiency symptoms are seldom seen in agronomic crops, but will typically include a failure of the new growth to develop properly. Annual grasses such as corn will have deformed emerging leaves that fail to unroll from the wool. The new leaves are often corotic. Extremely acidic soils can introduce an entirely new set of symptoms, often from different toxicities and deficiencies. Many fruit and vegetables demonstrate dramatic symptoms such as black heart in celery and broccoli, tip burn in lettuce and cabbage, white heart or hollow heart in cucurbits, blossom end rot in tomatoes and peppers, and pops in peanuts. Tree fruit with low calcium will exhibit increased storage problems such as bitter bite in the apples, cork spot in the apples and pears, cracking in cherries, and other degradation of the fruit while in storage. Deficiency in all crops often also impairs root growth and lead to additional symptoms as a secondary effect. Calcium deficient conifer trees will exhibit yellowing, then death and dropping of the needles of the new growth. The new growth may also be deformed. Calcium deficiencies can show as variety-specific symptoms. The image on the previous slide showed a calcium deficiency in a grapevine, while the image on this slide shows calcium deficiencies in strawberries. Calcium is found in the soil and available to plants in the calcium 2+ state. Calcium plays an important role in cell elongation and maintaining membrane structures in plants, and the presence of calcium in roots also regulates plant cation uptake by limiting excess sodium and increasing beneficial potassium absorption. Most soils contain abundant calcium in solution. That's about 30 to 300 parts per million, relative to most crop requirements, which is approximately 15 parts per million. The supply of calcium in the soil solution can be 10 times larger than potassium, and plants require much less calcium, so deficiencies are rare. Mass flow supplies adequate calcium to the plant roots, except in low calcium soils, where diffusion becomes an important process. Calcium is usually the dominant base cation in exchange reactions, accounting for more than 70% of the base saturation. Base saturation represents the percentage of the CEC occupied by the base cations, calcium, magnesium, potassium and sodium, and generally increases with pH. Exchangeable calcium exists in equilibrium within the soil solution, replenishing soluble calcium losses by plant uptake or leaching. Leaching can be significant in coarse textured acidic soils where substantial water moves through the profile. In many calcareous soils, some calcium leaches out of the more acidic, organic-rich topsoil and precipitates in the calcium-rich calic horizon in the subsoil in the form of calcium carbonate or gypsum. In addition to the dissolution of these secondary deposits, calcium is also released through the weathering of primary minerals such as feldspars, micas and limestone, all of which are common throughout many areas. Low calcium content in the soil often causes acidity problems before actual calcium nutrient deficiency becomes an issue. Both the role of calcium as the base cation and its frequent occurrence within carbonates, CO3, the bicarbonates, HCO3, buffer soil at higher pHs. Where acidity is a problem, limiting soils with calcium carbonate, calcium oxidate, and calcium hydroxide is a common practice. The role of pH in nutrient cycling and how it can be managed will be discussed later on. Because of its strong divergent charge, calcium acts as an ionic glue, attracting clay particles and promoting aggregation through a process called flocculation. Soils with high levels of sodium, referred to as sodic soils, promote dispersion, which is the opposite of flocculation. When monovalent, single-charged ions dominate the clay surface in the soil, the weak positive charge of the ion is strong enough to overcome the negative charges of the clay particles, which then repel one another. The result of dispersion is a structureless, gel-like soil with insufficient aeration, permeability, and water-holding capacity for optimal plant growth. Additions of calcium in the form of gypsum are frequently prescribed for reclaiming sodic soils because it counters the effects of sodium and promotes the aggregation critical for soil productivity. Gypsum is preferable to calcium carbonate because gypsum is more soluble. Low sodium irrigation water or rainwater can then be used to leach sodium out of the soil profile where appropriate. In conclusion, in the calcium cycle, we need to think about calcium, weathering and leaching, good soil organic matter, mineral and precipitates, and plant available nutrients. When we were learning about calcium and its role in plants, one of the functions noted was that calcium has interactions with other nutrients and substances in the soil. I am now going to investigate these in a bit more detail. Calcium is a major cation, and calcium availability in the soil is related to the CEC measurement. It is continuously in competition with other major cations, and these include sodium, potassium, magnesium, ammonium, iron, and aluminium. Calcium and sodium interact. When high levels of soil sodium will displace calcium and lead to calcium leaching. This can result in poor soil structure and possibly sodium toxicity to the crop. Conversely, applications of soluble calcium, typically as gypsum, are commonly used to desalinate sodic soils through the displacement principle in reverse. Phosphorus and calcium interacts, and this is pH dependent. As the soil pH is increased above 7, free or uncombined calcium begins to accumulate in the soil. This calcium is available to interact with other nutrients. Soluble phosphorus is an anion, meaning that it has a negative charge. Any free calcium reacts with phosphorus to form insoluble or very slowly soluble calcium phosphorus compounds that are not readily available to plants. Since there is typically much more available calcium in the soil than phosphorus, this interacts nearly always results in less phosphorus availability. As the pH of a soil decreases, more of these elements, iron and aluminium, become soluble and then are free to combine with calcium to form essentially insoluble compounds that is no longer available to the plant. And finally, calcium and boron interact to have negative implications on plant nutrition. High calcium or high soil calcium inhibits boron uptake and utilization. Calcium sprays and soil applications have been effectively used to help detoxify boron over applications. For many years there have been a few people who claim that there is an ideal ratio of the three principal soil cation nutrients, calcium, potassium and magnesium. This concept probably originated in New Jersey by work by Baer in 1945 that projected an ideal soil as one that had the following saturations of exchangeable cations: 5% potassium, 65% calcium and 10% magnesium, and 20% water. The cation ratios resulting from these idealized concentrations are calcium magnesium 6.5 to 1, calcium potassium 13 to 1, and magnesium potassium 2 to 1. It is generally accepted that there are some preferred general relationships and balances between soil nutrients. There is also a significant amount of work indicating the excesses and shortages of some nutrients, and how this will affect the uptake of other nutrients. However, no reliable research to date has indicated that there is any particular soil ratio of nutrients. Over the years, a significant amount of conversation and salesmanship has evolved around the concept of the ideal soil calcium-magnesium ratio. Most of the claims for the ideal ratio have been between 5 to 1 and 8 to 1. Some of the claims that are the correct soil calcium to magnesium ratio will improve soil structure, reduce weed populations, especially such weeds as foxtails and quackgrass, plus improve foliage quality. It is also claimed that it would reduce the leaching of other plant nutrients, generally improve the balance of soil nutrients. However, as I said, there have been much debate. Dr. Stanley Barber, for example, states there is no research justification for the added expense of obtaining a finite calcium to magnesium ratio in the soil. Research indicates that plant yield or quality is not appreciatively affected over a wide range of calcium-magnesium ratios in the soil. Winscombe research found that yields of corn and alfalfa were not significantly affected by calcium-magnesium ratios ranging from 2.28 to 1 to 8 to 1 in all cases. When neither nutrient was deficient, the crop's internal calcium to magnesium ratio was maintained within a relatively narrow range, consistent with the needs of the plant. These findings are supported by most authorities. A soil with the previous listed ratios would most likely be fertile. However, this does not mean that a fertile soil requires these specific values. Adequate crop nutrition is dependent on many factors other than specific ratio of nutrients. It will rarely be profitable to adjust the soil calcium-magnesium ratio. In later research, you can find references to nutrient ratios. However, in most cases, there will not be a specific numerical ratio associated with these relationships. The intention, rather, is to indicate that there is a relative abundance of the nutrients, and this changes significantly. It could affect the other uptake of other nutrients. This concept is much less specific and claiming that there is a value of a specific numerical ratio. Now let us have a look at some of the commercially available calcium fertilizers. Lime is calcium carbonate. The advantages of providing calcium in a fertilizer in the form of lime is that it allows for pH correction. The disadvantage is it's not as soluble and less reactive in non-acidic soils. Gypsum has the advantage of both calcium and sulfur nutrients and the flocculation of clays. Calcium fertilizers based on calcium nitrate have the advantages of non-acidifying nitrogen fertilizer. However, they are costly. Now let us learn about the secondary macronutrient magnesium. Magnesium is an essential plant nutrient. It has a number of roles and functions in plants. One of the best, or well known, is the critical role it plays in the photosynthesis process, as it is a binding block of chlorophyll, which makes leaves appear green and allows the light reaction of photosynthesis to occur. Other roles that magnesium plays: it is important in the production of ATP, adenylyl triphosphate, through its role as a coenzyme cofactor. It is also involved in nucleic acid formation and in respiration. Magnesium deficiency can result in intervenal chlorosis. Magnesium deficiency might be a single limiting factor in crop production. Magnesium uptake in plants is in the ionic form magnesium 2+. Uptake of magnesium is dominated by two processes. The first is passive uptake, driven by transpiration, and is similar to that of calcium. The second is diffusion, where magnesium ions move from zones of high concentration to zones of lower concentration. Therefore, the magnesium amounts that a plant can take up will depend on its concentration in the soil solution and on the capacity of the soil to replenish the soil solution with magnesium. Conditions such as low soil pH, low temperature, dry soil conditions, and high levels of competing elements, such as potassium and calcium, will reduce the availability of magnesium. Under such conditions, magnesium deficiency is more likely to occur. The effect of pH on magnesium availability is significant in acidic soils. The solubility of the magnesium decreases and it becomes less available. Due to the large hydrated radius of the magnesium ion, the strength of its bond to the exchange sites in the soil is relatively low. Acidic soils increase the tendency of magnesium to leach because they have less exchangeable sites, that is, a lower CEC measurement. In addition, in acidic soils, elements such as manganese and aluminium become more soluble and result in reduced magnesium uptake. Other positively charged ions, such as potassium and ammonium, may also compete with magnesium and reduce its uptake and translocation from the roots to the upper part of the plants. Therefore, excessive applications of these nutrients might prompt magnesium deficiency. Care should be taken especially in sandy soils or where your CEC value is particularly low. Magnesium soil content is at a preferred level in agricultural soils of 1.6 milliequivalents per 100 grams, or at a concentration of 192 mg per kilogram or more. The preferred calcium to magnesium ratio has been discussed. In soils, magnesium is present in 3 fractions. Magnesium in the soil is in equilibrium with the exchangeable magnesium and is readily available for plants. Exchangeable magnesium is the most important fraction for determining the magnesium that is available to plants. This fraction consists of magnesium held by clay particles and organic matter. It is in equilibrium with magnesium in the soil solution. And the third fraction is non-exchangeable magnesium. Here, magnesium consists of the primary minerals in the soil. The breakdown process of minerals in soils is very slow. Therefore, the magnesium in this fraction is not available to plants. You will not be surprised to learn that deficiencies in magnesium will lead to reductions in yield. It will also lead to an increased susceptibility of plant disease. Since magnesium is mobile, unlike calcium, deficiency symptoms appear on both lower and older leaves. The first symptom is a pale leaf, which then develops an interveinal corosis, as you can see from the grapevine image on the slide. In some plants you may see a reddish or purple spots. The expression of symptoms is greatly dependent on the intensity of which leaves are exposed to light. Deficient plants are exposed to high light intensities will show more symptoms. On the slide is another example of what magnesium deficiency symptoms may look like in the leaf. Magnesium, like calcium, occur in the soil as soluble divalent or double-charged cations, Mg2+, on cation exchange sites and in mineral forms. The major processes in the calcium-magnesium cycle are plant uptake, exchange, precipitation, weathering, and finally leaching. Calcium-magnesium dynamics in the soil are quite similar to that of potassium. Please see the lecture before this one, lecture 7. The most notable difference between these nutrient cycles is the absence of calcium-magnesium clay fixation. As mentioned previously, magnesium plays a critical role in nearly all parts of plant metabolism and protein synthesis and is an essential constituent of chlorophyll. Plants require less magnesium than calcium, but deficiencies are common because less magnesium exists in the soil solution. Mineral forms of magnesium are relatively resistant to weathering and represent a large fraction of the total soil magnesium. Mineral forms of magnesium include biotite, horny blade, and olivinin, dolomite, and most 2-to-1 clay minerals. Soluble magnesium can also precipitate out of the solution as magnesium carbonate and magnesium sulfate, frequently along with calcium carbonate in the subsoil surface. Although calcium and magnesium share the same exchange process, magnesium sorbs less strongly to the soil colloids and therefore is prone to leaching, particularly in sandy soils. As a cation, magnesium competes with calcium, potassium, and NH4 for plant absorption and cation exchange sites. Magnesium deficiencies occur when these other cations dominate soils with low magnesium concentrations, that is 10% of base saturation. A common magnesium deficiency problem in cattle is grass tetany. Due to the insufficient magnesium in the foliage, livestock can be fed magnesium salts in low magnesium areas, or soils can be amended with Epsom salt or dolomite additions. Because of the low solubility of dolomite at pH levels above 7, spraying dissolved or dry Epsom salts is most suitable for soils with high pH. There are several magnesium fertilizers commercially available. The most commonly used is dolomite. This has the advantage that it has both calcium and magnesium nutrients, and it allows for a pH correction. The disadvantage of dolomite is that it's relatively insoluble and slow to react. Magnesium oxide contains 54% magnesium. This is a high magnesium content, although, like the dolomite, it is relatively insoluble and slow to react. Potassium or magnesium sulfate fertilizers composed of 11% magnesium, 18% potassium, or 22% sulfur. The advantage of these fertilizers is they have a relatively soluble sulfate form of magnesium. However, these too dissolve slowly. We will now move on to the last section of this lecture and look at sulfur. We will start by looking at the roles and functions of sulfur in plants. Plants require as much sulfur as they do phosphorus. Sulfur is found in compounds such as cysteine, cysteine, methionine, amino acids that are required for protein construction. It activates certain enzyme systems and is a component of some vitamins such as vitamin A. Sulfur is found in mustard oil glycosides, which impact characteristic odors and flavors of such plants as mustard, onions, and garlic. Sulfur fertilization has been shown to increase the seed soil content of crops such as soybeans and flax. Plant animal scientists have shown that plant tissue content is typically 1 part sulfur for every 15 to 20 parts nitrogen, and that these ratios are required for optimal growth of animal feeds. It is essential in the manufacturing of chloroplasts. Sulfur is also found in the iron-sulfur complexes of the electron transport chains in photosynthesis. It is immobile, and deficiency therefore affects younger tissues first. Symptoms of deficiency include the yellowing of leaves and stunted growth. Deficiencies in sulfur can affect root growth and seed production as well as yield. The preferred level of sulfur soil content in agricultural soils is between 10 to 20 mg per kilogram. This does vary on the crop. Soil properties such as water content, pH, temperature, and aeration influence these processes and consequently affect the amount of sulfur available to satisfy any one crop's needs. Sulfur deficiencies are not that common in grapevines, but they do appear as chlorosis. Efficient nitrogen utilization requires adequate sulfur because both are needed to form proteins in the plant. Insufficient sulfur inhibits protein synthesis, causing nitrogen to accumulate in the plant as nitrate, amides, and amino acids rather than protein. Research has shown that sulfur plays an important role in crop qualities such as bread-making quality of wheat and the protein of forages and grains. Bread-making varieties of wheat have approximately 10% more sulfur in the grain than non-bread-making varieties, although total plant sulfur uptake is similar, as published in Rendig 1986 paper. Two important factors related to bread making are loaf volume and dough extensibility. Both of these factors are directly related to sulfur concentration in the grain, which is dependent upon available sulfur in the soil, as stated by Yunger et al. In 2002. As a constituent of amino acids, sufficient sulfur is essential for high protein content in foliages and grains. This slide depicts sulfur deficiencies in wheat. Sulfur undergoes numerous transformations in the soil, involving biological, chemical, and atmospheric processes similar to the nitrogen cycle. Only a small fraction of the total sulfur in the soil is readily available to crops. The major process governing sulfur availability in the soil are plant uptake, mineralization, immobilization, exchange, volatilization, precipitation, oxidation reduction, mineral weathering, and leaching. You will see this is quite a complex cycle and mimics that of nitrogen. Management of sulfur is improved by the understanding of how this nutrient cycle reacts in the soil and under what conditions deficiencies are likely to occur. Crops can uptake adequate amounts of sulfur in soils with soluble SO4 concentrations above the critical level of 5 to 10 parts per million in the top 6 inches of the soil via mass flow. Soluble sulfur usually represents less than 10% of total sulfur in the soil and is replenished by mineralization of organic matter, weathering of mineral sulfur, oxidation of reduced sulfur, and desorbation. A plant nitrogen to sulfur ratio of about 15:1 is thought of as ideal for effective protein synthesis. Ratios higher than this suggest that the protein synthesis is limited by a lack of sulfur, while smaller ratios imply surplus sulfur, which is retained as soluble sulfur within the plant, as reported by Stewart and Porter in 1969. In this way, nitrogen and sulfur interactions are positively related and should be managed together. No response to sulfur additions will occur if nitrogen is limiting plant growth. Similarly, an optimal sulfur level maximizes the effect of nitrogen fertilization on yield, although actual nitrogen uptake in the plant does not change, as cited by Rasmussen in 1997. Sulfur dioxide SO2 and hydrogen sulfide H2S from the atmosphere can also represent sources of sulfur for plants in locations downwind from coal-burning plants, metal smelters, geothermal areas, and urban areas. These compounds are beneficial only in low concentrations and are absorbed by plant leaves through stomata. Atmospheric additions to crops and soils range significantly, but they are usually about 2.3 to 10, as stated by Forth and Ellis in 1997. Crop residues contain considerable amounts of sulfur because this nitrogen is incorporated into the plant material and is not easily realized. As this is the case with other nutrients, the plant material removed at harvest can represent a significant loss to the soil ecosystem. In foliage crops, for example alfalfa, with this high sulfur uptake, most of the biomass is removed, resulting in a major loss of sulfur. Jackson in 2000 stated that in canola only about 5% of the total sulfur is removed with the harvested seeds, and so most of the sulfur remains in the residue. Therefore, this effect is crop specific. Tisdale et al. In 1986 showed that grazing foliages on site can significantly reduce sulfur losses because livestock release most of the sulfur back into the soil. In fact, it is stated as much as 90% of sulfur consumption by sheep is released back into the soil. I'm now going to talk briefly, specifically about mineralization and immobilization. You came across these processes with nitrogen. Organic sulfur compounds held in plant, animal and micro residues collect in the soil organic matter and represent the largest pool of sulfur in well-developed grassland soils. Over 90% of the total sulfur in these kinds of soils exists in the organic form, as stated by Haviland et al. In 1999, except in soils where mineral sulfur accumulations of gypsums are significant. Microorganisms decompose the organic matter and release plant-available sulfur through the process of mineralization, which occurs similarly in nitrogen cycling. About 1 to 3% of organic sulfur is mineralized each year, containing about 4.8 to 15.6 kilograms per hectare of soluble sulfur, SO4, for plant use annually. The amount of sulfur available to plants annually via mineralization processes depends on the amount of organic matter in the soil and the concentration of sulfur in organic matter. Therefore, taking steps to maintain or increase soil organic matter with such practices as no-till, minimum till, or organic additions can help supply a relatively consistent amount of available sulfur in your soil and allow your system to become sustainable. Immobilization refers to the process of converting inorganic sulfur, SO4, to organic sulfur, and is essentially the reverse of mineralization. Microorganisms remove available sulfur from the solution and convert it into proteins and other organic compounds. Although this process removes sulfur from the available pool, the sulfur is still in a reserve pool that could eventually become available to plants via mineralization. Since mineralization and immobilization are primary biological processes, any factors affecting microbial growth will influence both of these sulfur transformations. Important factors include soil temperature, water content, pH, carbon to sulfur ratio, aeration, and residual composition. The highest mineralization and immobilization rates will occur under aerated, warm, and moist conditions where near neutral pH levels, because these conditions are optimal for microbial activity. The nitrogen to sulfur ratio is relatively stable in organic matter, remaining near 8 to 1 as quoted by Forth and Ellis in 1997. But the C to sulfur, carbon to sulfur ratio is more varied and strongly affects the relative amounts of mineralization and immobilization. If residues and organic matter lack sufficient amounts of sulfur, microbes will pull the needed sulfur from the soil and immobilize it in organics, as in the case when a residue like sawdust is added to the soil. Residues with carbon to sulfur ratios higher than 400 to 1 will result in net immobilization of sulfur from the soil, while carbon to sulfur ratios lower than 200 to 1 result in net mineralization of soil sulfur. This was reported by Frenry in 1986. Between ratios of 200 to 1 and 400 to 1, a combination of the two processes may occur. The carbon to sulfur ratios for a number of organic residues can range. Each progressively decomposition cycle or soil residues results in carbon expelled as carbon dioxide, therefore lowering the carbon to sulfur ratio. In this way, even residues with high carbon to sulfur ratios can eventually supply the soil with available sulfur. In future farmings, when managing carbon dioxide levels, the relationship of carbon to sulfur could be critical. Biological volatilization can occur under both aerobic and anaerobic conditions, but is less significant in well-drained, well-aerated soils. Probably the most significant volatile loss of sulfur occurs when agricultural areas are subjected to fire. Although sulfate concentrations may increase due to chemical mineralization, that is, burning of organic sulfur, burning dry grass has resulted in losses of 75% of total sulfur in the vegetation cover, as cited by Tisdale et al. In 1986. The conversion of organic sulfur to inorganic sulfur by burning may also make the nutrient more susceptible to leaching. As you can see on the diagram, the components to think about when managing organic sulfur are decomposition, mineralization, weathering, and erosion. When sulfur is in the soil solution, it is present as an SO4 compound, a negatively charged ion. The processes that cycle with sulfur include uptake, immobilization, weathering, deposition, and erosion. Also, sorption precipitation will be considered. Inorganic sulfur occurs in solid phases in the soil as sorbid S or S minerals. Sorption of SO4 increases an anion exchange capacity, AEC, of the soil. Soils dominated by positively charged iron or aluminium oxide clays will have a high AEC and therefore sorb significant sulfate. In some regions, soils typically have low AECs, and there is a strong net negative charge and high pH levels, so sorption of sulfate is minimal. As a general rule, SO4 sorption is insignificant above a pH of 6.5. Inorganic sulfur minerals such as gypsum represent an important sulfur pool in some soil communities. Commonly, gypsum accumulates in the subsoil, forming a sulfur-rich layer, often in close proximity to calcium carbonate layers. Although the surface layers of these soils may have low levels of plant-available sulfur, the deeper mineral sulfur maintains an adequate supply of sulfur within the rooting zone depth of most crops, and this was referenced by Beaton and Soper in 1986. Therefore, your soil sampling needs to take into account this effect. Early in the growing season, subsoil sulfur may not be available to crops because it is out of reach of the growing crops. Later on in the growing season, when soil water is usually moving up and down the profile and plant roots have grown deeper, adequate sulfur is generally accessible to crops. Sulfur occurs in numerous primary and secondary minerals, which release either sulfate or sulfide, S2, as they weather. Gypsum is widely distributed in acid and semi-arid soils, where precipitation is too low to leach the mineral out of the profile. Shales and claystones in some regions contain gypsums and release plant-available sulfur as they weather. Ore deposits can contain metal sulfides such as iron, zinc, and manganese sulfides. As the sulfide, S2, are exposed to oxygen, and the water they oxidize to sulfates can significantly lower the pH of the surrounding soil or water. This is as in the case with acid mine tailings. In agriculture settings, the slow release of sulfur minerals in the soil can be beneficial, but excess acidity may be a concern. Elemental sulfur also oxidizes to sulfate in the presence of water and air and is commonly applied to the pH soils, both as sulfur fertilizer and as a means of lowering pH for plant growth. The resulting acidity is generally short-lived in well-buffered soils and requires contained application to be maintained. Gypsum added to the soil as a fertilizer will neither raise nor lower the pH of the soil because the sulfur is already completely oxidized in the form of sulfate. Sulfides and elementary sulfur are found in waterlogged soils and wetlands and generally exist in low concentrations in most agricultural soils. Sulfur is lost from the soil by two processes other than crop uptake, leaching and volatilization. Leaching is the physical removal of the plant-available sulfur by water moving through the profile, whereas volatilization represents a biological or chemical transformation and subsequent release of sulfur into the atmosphere. The areas with the highest risk for sulfate leaching are associated with high precipitation, irrigated conditions, and coarse textured shallow soils, and soils with low EC. Irrigation following SO4 fertilizer application can move SO4 through a profile and eventually out of the reach of plant roots. Sulfate leaching represents an economic loss because once the sulfate has left the soil system, it is no longer available for the crop uptake. In semi-arid climates, sulfate often collates in the subsoil as described earlier because there is insufficient water to flush the anion below the rooting zone of plants. Leaching losses are generally less in the high pH soils because sulfate will precipitate with calcium or magnesium. It should be noted that in some soils, water moving upward through the soil profile via capillary action late in the growing season can carry sulfur towards the plant roots and the soil surface. In such instances, excessive calcium and magnesium sulfate salts can accumulate in harmful levels in the surface and limit plant growth. Careful water management, especially in irrigated cropping systems, such as those found in viticulture, can prevent many of the problems associated with the leaching of sulfur or the overaccumulation of sulfur salts. Fertilization of sulfur compounds represents a relatively small loss in most agricultural soils. Generally, the amount of sulfur volatilized from the soil is less than 0.05% of the total sulfur. From plants, the release is between 0.3% and 6% of the total sulfur in the plant, as reported by Haviland et al. In 1999. Most volatilizations in soils occur during bacterial decomposition of organic residues. There are three commonly used sulfur fertilizers. Sulfate of potash, dipotassium sulfate, contains 18% sulfur and 41% potassium. The advantage of using this fertilizer is that it contains both sulfur and potassium. The disadvantage is that it is more expensive than muriate of potash. Ammonium sulfate contains 24% sulfur and 20% nitrogen. It is advantageous as it has both sources of sulfur and nitrogen. However, it is usually supplemented with other nitrogen fertilizers. And finally, gypsum, calcium sulfate, contains 15% sulfur and 90% calcium. This has the advantages of having both calcium and sulfate nutrients, and flocculation of clays. And this brings us to an end of Lecture 8 on secondary macronutrients. You have learnt about plant function and the role each of these nutrients will play. You have learnt about the different cycles. Some are similar to nitrogen, while others are relatively simple. We have looked at examples of commercial fertilizer for each of these compounds. When reviewing this lecture, please ensure that you compare and contrast the differences of these macronutrients. This will complete your notes on this topic.